Welcome back to Bio 111. This is chapter 16, section 5. We're looking at post-transcriptional gene regulation. All right, so unlike prokaryotes, the RNA that is created, that is transcribed immediately from the DNA, is not ready to be turned and to be translated into protein. We have to go through post-transcriptional modification. All right. This is another step in which we can assert some level of control. All right, so the pre-mRNA. So we've been calling the RNA that comes straight from the polymerase during transcription, we've been calling that mRNA, but in reality, it's really pre-mRNA. Because sitting inside of that RNA are these sections of DNA called introns. Now, introns are DNA that will not be transcribed. We'll take them out and basically throw them away. Right? We call this splicing. Right? So we're going to take the introns out and leave just what we call exons. In this case, in this picture, those are the 1, 2, 3, and 4 segments. If you see here, we've got two different sequences. We've got 1, 2, 3 and 1, 3, 4. We can splice these together in alternative patterns to get different proteins. Another level of control here. This is just a little cartoon showing these different types of splicing patterns that can result from different introns being taken out and different exons also being removed. All right, such as exon skipping, right? Just completely skipping over an exon and letting it leave the sequence as well. All right, I just want to remind you, we're not taking DNA out. We're not altering the genome in any way. We're altering the mRNA, the pre-mRNA that was transcribed in the nucleus and has now come out. All right, how we modify the RNA at this point is going to determine what the proteins look like. All right, another way in which we can regulate gene expression is actually through the stability of RNA. All right, one thing to remind you, we have a lot of RNases in our cells that will degrade RNA. We have a lot of RNases on our skin that will also degrade RNA. If you're working in a lab that works with RNA, you're doing a transcriptional study, you're going to have to spray down your bench all the time and clean your hands constantly to get rid of these RNases because they will degrade RNA. Now, the reason why we do that, the reason why we have so much RNase is likely to help with some of this RNA control here, and also because a lot of viruses use RNA to infect us. So it's a good defense mechanism. All right? But part of the control of RNA is how stable it is. How long will it stay around and be able to be translated into protein? If you think about it, if you've got an RNA that gets put into the cytosol, the cytoplasm, stays around for a little bit, very briefly, only has 2 protein molecules synthesized, translated from it, it's not all that effective, right? But let's say that you've got a RNA molecule that comes into the cytoplasm and it stays around for a long time and has thousands of protein molecules translated from it. That's a very effective, very prolific RNA at that point, right? So there is a level of control. And the way that our cells control the stability is by protecting the RNA, right? They put a 5' cap end, a 5' cap on the ends. This is actually a guanosine triphosphate molecule, kind of the brother of ATP. This is GTP. It binds to the 5' end of this RNA, prevents degradation from happening. And then we have a poly A tail that are added to the end of the mRNA. And this poly A tail is, in a lot of ways, it's kind of sacrificial material that prevents degradation of important coding DNA, that is, or RNA, that is RNA that will be translated into a protein. You'll also see that there are these UTRs on either ends, the 5' UTR, 3' UTR. These are the untranslated regions. And these act kind of like tags. They tell the RNA where it's going to be going in the cell for further processing. Now, there are some other types of RNA that we haven't talked about yet, and they're really small. They're short RNA molecules, 21 to 24 nucleotides long. They're called microRNAs or miRNAs. Now, they recognize specific sequences of mRNA. That is, they will bind to it. Now, when they bind to it, they can associate with what are called a RISC complex. Now, RISC stands for a ribonucleoprotein or RNA-induced silencing complex. Now, silencing, gene silencing, is when you basically turn off that gene, or the expression of that gene, by degrading the mRNA. And that's what's going to happen here. This miRNA, this microRNA, binds to its specific section of RNA, of mRNA, and induces its degradation, all right, and thus silences it. You see this in some plant studies. So one technique in trying to understand how genes are regulated inside of plants is you tag them with a fluorescent protein that you can see turn on and off. Now, one problem that you might have is if you tag too many different genes with these proteins, because if you have too many of these genes tagged, the microRNAs, these RISC complexes, will come in and silence your protein of interest, and you won't see anything happening. So you got to be very careful. So this does come up in experiments. All right, so that is section 5. We're going to be talking about section 6 in the next video.